Okay, so far what we've covered is the process of DNA replication in bacterial cells, and now we're going to get into a little bit about how it works in eukaryotic cells. So they share many features. So the double-stranded DNA is unwound at a single point, or at a point called the origin. A replication fork forms. Bidirectional synthesis creates leading and lagging strands. And then eukaryotic polymerases require 4 DNTPs, templates, and primers. So a lot of that fundamentals is the same. But there are differences. There are very important differences. And 3 of them that you should remember are: one is that there's way more DNA in eukaryotic cells than prokaryotic cells in general. So we talked before about E. coli bacteria, and that has 4 megabases of DNA. Humans have approximately 2 gigabases of DNA. So it's almost 1,000-fold more DNA that actually has to be replicated. And so that presents a challenge in how you can actually replicate that much DNA in a short period of time. They also have linear chromosomes. So bacteria cells have circular chromosomes. Eukaryotic cells have linear chromosomes, and that actually presents some problems for how to replicate the ends of chromosomes. And then finally, DNA is complex with nucleosomes, so DNA is wrapped up around nucleosomes, and so that also has to be dealt with. So how do they deal with the challenge of replicating so much DNA? So we'll think about why this is so, but instead of having a single origin of replication, so in the bacteria we started from one end of the circle and then we replicated to the point at the end. Eukaryotic chromosomes contain multiple origins of replication. So a single chromosome, which is 180 megabases long, it's initiating from multiple different spots within there. And that facilitates the rapid synthesis of a large quantity of DNA. So because it's synthesizing from so many points, you can actually synthesize it faster. And so you can actually see kind of examples of this. So this is a picture. This is an EM micrograph that was taken. And you can see approximately how long 5 kilobases is within that. And I find the schematic a little bit easier to kind of pay attention to. But you see all sorts of replication bubbles. So here is an example of a replication bubble. And so there, at some point there was an origin of replication right in the middle, and then it proceeded bidirectionally in both directions. And so now you see this bubble. This is another bubble right here. And so there was an origin of replication somewhere in the middle, and then it proceeded bidirectionally to create this bubble. And so all along this entire chromosome, you can see examples of all of these different bubbles. And so eventually these bubbles will meet together and then fuse into the double-stranded DNA, the 2 new pieces of double-stranded DNA. So DNA polymerases involved in nuclear genome DNA replication. So there's the polymerases alpha, delta, and epsilon. Those are involved in initiation and elongation. The alpha polymerase possesses low processivity, so it doesn't stay on the strand very long. And then RNA primer synthesis during initiation on leading and lagging strands. So these are some of the DNA polymerases that are used in eukaryotic cells. So the alpha creates the RNA DNA primers and the initiation of DNA synthesis. Delta and epsilon are the primary polymerases used for replication. These are like DNA polymerase 3 for bacteria, but they're actually separated into leading and lagging strand synthesis. So unlike bacteria, which use the same polymerase for both the leading strand and the lagging strand, eukaryotic cells use different polymerases, the delta versus the epsilon. There's also a special polymerase used for mitochondrial DNA replication and repair. So mitochondria are the energy source of the cells, but they also contain their own genomes. And so they have a special polymerase that's used for replicating their genomes. And then there's also a bunch of polymerases used for DNA repair, so different types of DNA repair, base excisions, translesion, etc. So polymerase switching occurs once the primer is in place. So the alpha, polymerase alpha, and epsilon are replaced by polymerase D for elongation. So the polymerase delta synthesizes the lagging strand, and the polymerase epsilon synthesizes the leading strand. You also have to deal with replication through chromatin. So here is a picture of a replication fork. So you have the old strands coming in this way, and then you have the new strands coming in this way. And so you notice that these are actually decorated with nucleosomes. And so this presents two problems. So one is that you have to be able to replicate through the nucleosome. So if the nucleosomes are still there, they might stall and stop the polymerase because the DNA is wound around them. And then you also notice that you actually have to replace nucleosomes on each strand. So you have to not only duplicate DNA, but if you want to keep the organization of the DNA the same on both of the new strands, then you also have to add the nucleosomes back. And so the cell has a mechanism for that. I won't cover this in much detail for the exam. I just want to give you some idea of these things. But what I will cover and what I really want you to know about are the telomeres. So telomeres are the inert chromosomal ends that protect eukaryotic chromosomes from improper fusion or degradation. And these are long stretches of short repeating sequences that preserve the integrity and stability of the chromosomes. So telomeres are important both for the ends of the linear chromosome, but they're important both for protecting it, but then also for actually replicating and properly replicating the linear chromosome. And so why do we talk about this? Why are we worried about this in a chapter on DNA replication? And that's because if you think about what happens when you're doing DNA replication on a linear chromosome, there's clearly going to be an issue. With the ends of chromosomes. And so here we have these two strands. These two strands are going to provide the templates, and these are going to be synthesized. These are going to provide templates to synthesize new strands. And so we have a leading strand. So the leading strand, of course, just continues on over and over in the 5' to 3' direction. And then you have the lagging strand that does this in a discontinuous fashion. So, the replication fork is moving in this direction for both of these. So, things are going well. RNA primers are created. They are then removed. Gaps are created, and then these can be filled in. So these are filled in starting at the 3' end of a strand and moving towards this right here. So the problem with the ends of the chromosomes is that there's not actually, once you remove that RNA primer, there's not actually something in order to start polymerization from. And so you're going to have this gap right here and this gap right here in both of the newly synthesized strands. And so if this continues on, generation after generation, you're going to progressively shorten the DNA until, you've lost all of the DNA. So this is obviously a problem. The cell can't just let this happen. The cell has to come up with some sort of mechanism that allows the ends of the chromosomes to be synthesized as well. And so this is where this enzyme telomerase comes in. So telomerase is not the same as telomeres. So make sure you realize that these are two different words. Telomeres are actually sequences of DNA. Telomerases are enzymes or proteins that serve as, that have a catalytic activity that allows for replication of the ends. And so these are actually interesting. These are ribonucleoproteins. So these are both proteins, but then they also contain RNA, pieces of RNA within it. And so this RNA is actually serving as sort of a new primer in order to elongate the ends of DNA. So this kind of summarizes how it works. So we had DNA replication go pretty well, and we have a set. So the telomeres, you'll notice, are repetitive in nature. So this TTGGG, TTGGG, and then we start with the TTG right here. So this is what we mean by telomeres are repetitive in nature. They're a repeat of very short sequences. So here we've drawn them as 6 base pairs. And so these telomeres are actually complementary to the telomerase that includes the RNA molecule within it. And so the complementariness means that it has to be AACCCC, AA, instead of TTGGGGTT. But this complementariness allows the RNA within the telomerase to bind to the telomere. Once this telomerase binds, we are able to synthesize in this direction. So the DNA polymerase is actually, the telomerase is actually able to synthesize on the strand to add complementary nucleotides to the RNA molecule. So this is an RNA-dependent DNA polymerase activity. It's using an RNA as a template, but it's able to synthesize new DNA. And so this is able to, add, let's say, 6 base pairs of DNA. And so now this, once it reaches the end, the telomerase is actually able to translocate. And so this AAC right here can move to be complementary to the TTG at the end. And so you're able to continue this process and actually lengthen the ends of the DNA, the linear DNA. So we've synthesized this kind of over and over again. So now, when a normal replication occurs, we're dealing with a longer piece of DNA. So we've extended the length of the old strand, and so now primase is able to come in and synthesize a new primer, an RNA primer, that's able to add DNA to the newly synthesized strand. So from this primer, DNA polymerase goes in the 5' to 3' direction this way. Now the primer is removed, and so the ends of the newly synthesized DNA is actually shorter than the old one, but because we've lengthened this first, this completely synthesized one is longer than what was there before. And so we're not, so you will note that it is a little bit shorter than before. It is shorter than the template strand, but what's most important is that it is not shorter than the original size of the template strand. So this piece right here on the newly synthesized piece is actually longer than this initial piece. And so the action of telomerase is actually able to extend these ends and prevent the progressive shortening from occurring over time. So this is actually, so if we have DNA replication, the only way that shortening doesn't occur is if telomerase is active. So this provides a way of regulating the cell, because they can also regulate the presence and activity of telomerase. So actually, in most eukaryotic somatic cells, if you just grab some random cell within your body that's duplicating, the telomerase is not actually active. And so in those cells, the chromosomes, the linear chromosomes, are actually shortening. In your germline cells, for example, the telomerase is active to make sure that what gets passed on into your gametes is the properly sized linear chromosomes. But because of this, so stem cells, for example, these are the cells that constantly renew, or the source of renewal for cells. These maintain telomerase activity, and this maintains the integrity of the DNA. And this is actually also important for cancerous cells. So if you're interested in cancer, tumors develop, and these tumors are actually just replicating, your cells kind of replicate over and over and over again in an uncontrolled manner. And so if these malignant cells didn't contain mutations or some other reason to increase telomerase activity, eventually these tumor cells would die because of the progressive shortening of the DNA chromosomes. So that's one of the factors that's thought to be important for the evolution of cancer, is that they're actually able to activate the telomerase activity to prevent that. And so that's why this activity and the telomere length is linked to things like aging, cancer, and other diseases. So we've covered, a brief set of things of eukaryotic chromosome and DNA replication. Obviously, there's many more things, or hopefully you appreciate that there's many more things that are going to be different. We've just touched on some of the key aspects of that. So hopefully that all makes sense, and now we will move into recombination and DNA repair.